So barley researchers, they do a really good job of breeding varieties that are wanted by our end-use customers, whether it's in China or Japan or otherwise. And we really rely on them to come up with varieties that are not only high yielding, but they also have good standability and have very good disease resistance packages.
We usually tend to use elite parents that we cross, and they are complementary to each other. For example, we have one parent that has good yield and another parent that has not so good yield but good quality. We cross these two parents with the aim that the offsprings will have the traits, both traits of the parents, good yield and good quality. At Brandon, we take pride on our focus, strong focus on diseases. The center is the host of 3 disease nurseries that are led by my colleague James Tucker. And those diseases are priority 1 diseases. So that will help us to ensure that the breeding lines that we are developing have resistance to those important diseases such as leaf diseases, stem rust, or Fusarium hand blight.
Some of the research that I'm involved with is identification of new resistant sources, so screening gene bank collections and looking for new resistant sources from all reputed sources from international people working in barley. So identifying genes that we can incorporate into the breeding program. So once we've identified these, they make them available to the barley breeders who can make crosses. Once crosses are made, we can put them into our disease nurseries and screen them for looking at lines that carry resistance with agronomics and acceptable malting quality.
Disease-resistant varieties are your foundation in terms of disease management. And if you can use a disease-resistant variety with a high level of resistance, that really mitigates your risk. So it provides some peace of mind for the farmer. They don't need to look at added input costs in terms of fungicide application or applications within a growing season. We work as part of the team with the breeders and we screen their breeding material, or in some cases they may have lines of barley they've brought in from other programs that they want to see if they have effective sources of resistance that they can incorporate into their breeding programs. So we plant out thousands of breeding lines for the programs. They send that material to us. We then inoculate that material with either old infested straw from the previous year, so it could be scald infested straw or the form of net blotch infested straw or so on. And then we also, in some cases, with scald, we inoculate with a spore suspension of the canidia of the fungus, or in the case of the net blotch pathogens or spot blotch, we inoculate with what's referred to as grain spawn inoculum. So it's basically taking kernels of, could be corn in the case of Fusarium graminearum and Fusarium head blight, but in our case it's mostly winter wheat. We autoclave that, grow the fungus on it, and then we spread that material in the field.
In the more recent past, people have been using DNA markers in a very, very specific fashion, looking to select for a specific gene, giving resistance to a disease, for example. Nowadays, with these broader genetic fingerprints, we're able to really do a more larger coverage, a bit like people when they do their Ancestry.ca thing. They're getting a lot of information that allows them to say, oh, you're 25% Irish, 50% Scottish, and so on and so forth. Well, plants have some ancestry of their own, and in doing these genome-wide markers helps us understand what the relationships are between these different varieties and lines that we have in our programs. So one of the main goals of the CropSnip project is affordability of the technology. So currently, if a breeder wants to do the type of testing, genetic testing on the scale that we're talking about, the cost will be probably in the $50 to $100 range. And we're looking to bring down this cost below $5 a sample. And so I think that by making these tools more affordable, we increase their use and expand their use also into new areas. And I think for breeders, this is some new tool that will be easier to adopt, because breeders typically work on very, very large sets of materials, tens of thousands of lines. And so you can imagine that if it costs $100 a line, it quickly becomes completely unaffordable. And so keeping that cost low is really a very important driver.